I'm Joe Graydon.
And I'm Terry Graydon. Welcome to this podcast of the People's Pharmacy.
You can find previous podcasts and more information on a range of health topics at peoplespharmacy.com. How well do you feed the bacteria in your belly? The food you eat affects the health and diversity of your microbiome. This is the People's Pharmacy with Terry and Joe Graydon.
Until recently, most people thought of microbes as germs to be avoided. It turns out that we are hosts to trillions of bacteria, viruses, fungi, and other organisms that influence our mental health, immune system, and general well-being.
The typical American diet does not support a thriving and diverse microbiota. Antibiotics can wreak havoc with the population of bacteria living in your intestines.
Coming up on the People's Pharmacy, find out how the microbiota in your digestive tract is taking care of you.
First, the news.
In the People's Pharmacy health headlines, many parents of young children are pleased that the FDA has been asked to consider emergency authorization of vaccines for kids under 5. The request from Pfizer-BioNTech is unprecedented. Although the vaccines are effective in children from 6 months to 2 years old, they're not as effective in youngsters between 2 and 5. Pfizer is currently conducting trials of a third dose in this age group to see if it can bring the immune response up to speed. Rather than wait until the trials are complete, leaving babies and preschoolers completely unprotected for a few more months, federal regulators want to give parents a chance to get kids vaccinated sooner. The idea is that even partial protection is better than no protection. Dr. Paul Offit, who serves on the FDA's Vaccine Advisory Panel, has questioned the wisdom of authorizing a vaccine that is not fully effective.
How good are rapid at-home tests for COVID-19? A Canadian study published in JAMA found that the rate of false positives was very low. If a person tested positive on the 15-minute antigen test, they were immediately asked to undergo a PCR test that's considered the gold standard. There were over 900,000 rapid antigen tests, resulting in 1,322 positive results. The false positive rate was 0.05%, and a number of those resulted from a single bad batch from one manufacturer. The researchers point out that rapid home tests can identify contagious people with reasonable accuracy. Those who are negative can go back to work without much fear of spreading the virus.
One of the more promising advances in cancer treatment was introduced several years ago. It's called chimeric antigen receptor, or CAR, T-cell therapy. Another description is the adoptive transfer of T lymphocytes, reprogrammed to target tumor cells. In this therapy, patients with blood cancer, such as leukemia or lymphoma, have their own T-cells harvested. They then undergo laboratory manipulation to produce modified T-cells to fight the cancer. These CAR T-cells are then reinfused back into the patient where they begin to attack the cancerous cells. A question that's challenged researchers is, will this unique immunotherapy last? A small study just published in the journal Nature suggests it will.
In 2010, 2 patients with chronic lymphocytic leukemia were treated with CAR T-cells as part of a Phase 1 clinical trial. The researchers now report durable remissions a decade later. Both patients remain cancer-free. What has these investigators especially excited is that the CAR T-cells seemingly evolved to seek out and kill tumor cells that themselves may have changed over time. The downside is that CAR T-cell therapy can cost up to $500,000 or more, and adverse reactions can be severe. Cytokine release syndrome can produce symptoms such as fever, fatigue, headaches, nausea, vomiting, body aches, rashes, irregular heart rhythms, low blood pressure, confusion, seizures, and breathing difficulties. All that said, the long-term anti-cancer benefits of this living cell therapy are impressive.
Autoimmune conditions can be hard to treat. They include disorders such as polymyalgia rheumatica, rheumatoid arthritis, psoriasis, and thyroid disease. These illnesses are not only uncomfortable, they're also a leading cause of death, especially in women. A new study published in the BMJ reports on the effects of vitamin D and omega-3 fatty acids on these autoimmune conditions. Over 25,000 participants were randomized to receive 2,000 international units of vitamin D, or a look-alike placebo, or 1,000 milligrams of omega-3, or a placebo. They were followed for more than 5 years. At the end of the trial, the researchers found that daily supplementation with either vitamin D or omega-3 fatty acids reduced the likelihood of developing an autoimmune disease. According to the authors, supplementation with both vitamin D and omega-3 fatty acids decreased autoimmune disease by about 30%. And that's the health news from the People's Pharmacy this week.
Welcome to the People's Pharmacy. I'm Joe Graydon.
I'm Terry Graydon. We humans are habitat to trillions of microbes. In fact, there may be more bacterial and viral cells in our bodies than there are human cells. But although we've long thought of microbes as germs, as our enemies, it turns out that the collections we host are crucial to our health and well-being. What should we know about our microbiota?
Our guests today are a husband and wife research and writing team. Dr. Justin Sonnenberg is an associate professor in the Department of Microbiology and Immunology at the Stanford School of Medicine. Dr. Erica Sonnenberg is a senior research scientist in the Department of Microbiology and Immunology at the Stanford School of Medicine. She studies the role of diet on the human intestinal microbiota. They're co-authors of The Good Gut, Taking Control of Your Weight, Your Mood, and Your Long-Term Health.
Welcome to the People's Pharmacy, Dr. Justin Sonnenberg and Dr. Erica Sonnenberg.
Great to be with you.
Yeah, thanks for having us.
In the introduction to your book, Dr. Andrew Weil notes that when he went to medical school, there was no concept of the microbiome, or at least nobody called it that. The idea that bacteria and fungi and viruses in the digestive tract could influence the rest of the body was pretty foreign. People who ate yogurt or took acidophilus supplements, they were kind of a little weird. How has all that changed in the last couple of decades?
Yeah, there's been this awakening that we, harbor an incredible microbial community all over our bodies. Most of these microbes live in our distal gastrointestinal tract and are known as the gut microbiota or the gut microbiome. But the amazing part of this is that we're realizing that these microbes are just a integral part of human health. And for decades, we've been declaring war on microbes. And now we realize that not only are not all microbes bad and disease-causing, the vast majority of microbes we encounter are actually beneficial to our health. And so it's just led to this total change in how we think about all of these little microorganisms that are everywhere.
And everywhere, really. Today we're going to really focus on the digestive tract, but I think we have microbes in any conceivable human space.
Yeah, microbes are amazingly adaptable, and so they can colonize virtually almost any environment. So anywhere that there's, a spot for them, they will figure out a way to survive. So most of the bacteria that we harbor live in our digestive system, but there are bacteria, on our skin, in our belly button. I mean, anywhere you can imagine, ears, eyes, all those places have microbes.
In The Good Gut, you speak of the microbiome as a second genome. Could you explain, please, what you mean?
Yeah, so there's been, a tremendous focus on the human genome over the past couple decades, and it's been justified. I mean, we know that the human genome contains the blueprint for each one of us in terms of our biology. But what the study of the gut microbiome has taught us is we've turned this DNA sequencing technology away from the human genome and towards these microorganisms, is that the vast majority of genetic material that each human harbors is not in our human genome, but it's in these resident microbial communities. And so there's just a tremendous capacity for these microbes to influence and dictate our biology. It's estimated that the human genome has around 20,000 genes. Our gut microbiome alone has, each one of us has on the order of 100 to 1,000 times the number of genes as our human genome. So just, it greatly eclipses the diversity of biology that can occur.
One interesting thing about that is you think of, you're born with your human genome, which is static. There's not really much you can do to change that. But your genome that you get from your microbial communities is something that we actually have a lot of control over based on our lifestyle or diet choices. And so it's a really powerful message to think that, 99% of your associated genetic material is something that you can actually have some control over and improve your health through.
That is extraordinary. And I guess, we always hear from people these days, oh, yeah, there are more bacteria in our digestive tracts than there are people on the planet. And then I read on your in your book that there are more microbes on our hand than people in the world. And I'm going, wow, really?
Yeah, it totally changes how you think about your body. You think of humans as a single species, Homo sapiens, and we're actually a composite of hundreds to thousands of species. So we are a walking ecosystem. It's like each one of us is a rainforest in terms of the diversity of life. And then if you start counting the actual cells, microbes are just way more abundant in our body, the microbial cells, than the human cells. And so by cell number, each one of us is actually more microbial than human. And so there's kind of a evolving view of when you look at somebody, what you're actually looking at. It's not just a human, it's this composite organism with microbial and human parts that come together to work in an incredibly elaborate way.
Now, when I was growing up, it seemed like, as you suggested, there was a war with microbes. I remember a commercial on television where Listerine was supposed to kill germs by millions on contact. And then we have tabletop cleansers that say they'll kill 99% of the germs and viruses that cause illness. And so we've been fighting all of this stuff for years. We wash our hands in antimicrobial soaps, and we are fearful there are germs, bad germs, just lurking everywhere to attack us. You're suggesting we should rethink that concept.
Yeah, I mean, it's interesting. Microbiology started out as an endeavor to understand the bacteria that cause us harm. And, with the advent of antibiotics, it was really very successful in making people well. And antibiotics saved, countless lives. And so, those have been amazing drugs. But as we understand more and more about this microbial world that we live in, there's been a realization that there are bad germs out there, but there are many, many more examples of good microbes. And this sort of carpet bombing that we've done of microbes through the use of antibiotics and all these cleaners and antimicrobial soaps has really done a disservice in a way, in that it's not just removing the bad microbes, but also the good ones. And so we just need to rethink our relationship with microbes and understand that there are very beneficial microbes that we have to care for, because they're really a part of who we are. Like Justin said, this walking ecosystem that we are.
You've emphasized the importance of diversity of our microbes. Why is that so important?
It's an excellent question, and I think, the field of microbiome research is maturing at a rapid rate. The number of discoveries are mind-boggling, but diversity is one of these things that we really still don't understand very well, except for just kind of general ecological principles. And it really appears that whether you're looking at a rainforest or whether you're looking at a gut microbiome, in general, there is a relationship between the diversity of that community and the health of that community. Now, there are ways in which diversity can get out of control, and there can be too much diversity or the wrong type of diversity that actually is disease causing. And this is, where the nuance in the field really comes into play is understanding what optimal levels of diversity are and what the right types of diversity are. But what is very clear is that most people that live in the industrialized world that have been exposed to antibiotics and sanitation and this kind of hyper hygienic society, and then also consume a Western diet, have a gut microbiome that is probably lacking in the proper amount of diversity for optimized human health.
Now, I'm trusting that there are ways that we could change that. And we'll talk more about that in a little bit, but maybe you could just describe for us how a baby gets started with its microbial diversity, and then how do we change that as we go on?
Yeah, so when, a baby is born, the womb is largely a sterile environment, and so they enter the world pretty devoid of microbes. But upon birth, they enter this, microbial world that we all inhabit, and they're rapidly colonized with microbes. And depending on various situations, the first microbes that colonize that baby will be different. So, for example, if that baby is born vaginally versus by C-section, that will influence the type of microbes that infant is first exposed to. Whether or not that infant is then breastfed versus formula-fed will also influence the first communities that assemble on and within that child. And there's growing evidence that these early colonizers are important in training this infant's immune system. So when you're born, your immune system is still not fully developed and requires cues from microbes to fully mature. And so which microbes first colonize that infant can send that child on a trajectory toward their ultimate immune system that they end up with. And there's evidence that sanitation, C-sections, formula feeding perhaps prime that child's immune system in a way that makes them more susceptible to many of the diseases that we see going up in the West, things like asthma, allergies, and other autoimmune diseases.
You're listening to Dr. Justin Sonnenberg and Dr. Erica Sonnenberg. He is an associate professor and she is a senior research scientist in the Department of Microbiology and Immunology at the Stanford University School of Medicine. Their book is The Good Gut: Taking Control of Your Weight, Your Mood, and Your Long-Term Health.
After the break, we'll find out how the microbiome interacts with the rest of the body.
Can the denizens of our digestive tract really affect big health problems like inflammation, insulin resistance, and obesity?
Find out what scientists know about how to change the balance of bacteria in our guts and what they're still struggling to learn.
If we could reprogram our microbiota, could we treat irritable bowel syndrome or even inflammatory bowel disease?
We'll find out how behavior and cognition may change with alterations in the microbiome.
You're listening to The People's Pharmacy with Joe and Terry Graydon. Welcome back to The People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon. If you would like to purchase a CD of this show, you can call 800-732-2334. This is show number 1156. That number again, 800-732-2334, or visit our website, peoplespharmacy.com. You can also download the free podcast from iTunes.
The People's Pharmacy is brought to you in part by Verizana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome. Online at verisana.com.
Today, our topic is intestinal microbiota influences on our health.
We're talking with Dr. Justin Sonnenberg and Dr. Erica Sonnenberg. He's an associate professor, and she is a senior research scientist in the Department of Microbiology and Immunology at the Stanford University School of Medicine. Their book is The Good Gut, Taking Control of Your Weight, Your Mood, and Your Long-Term Health.
I've been wondering how the microbiome interacts with the rest of our body. You've mentioned allergy and immunology, but what about things like inflammation or obesity, insulin resistance, even our brain? So could you give us a quick overview of how all those bacteria and presumably other things as well, fungi, viruses, and goodness knows what else, may be interacting with us?
Yeah, it's a great question. And I think, for decades, it's been apparent and intuitive that the gut microbiota would influence digestion of food, would influence gut health. But what has really been remarkable in this field is to watch all of the examples accumulate of how our gut microbes can influence this diverse array of aspects of human biology, human health, that are what we think of as separated from the gut, can influence moods, behavior, immune system in the lung, metabolic aspects of our health, obesity, and really the experiment that I think got a lot of people's attention, which occurred over 10 years ago now in a lab where Erica and I were doing postdocs. It was an experiment performed by somebody else, but it was in Jeffrey Gordon's lab at Washington University in St. Louis. They first observed that a microbiota of an obese mouse was different than a lean mouse. And so the question became, well, is that different microbiota a cause of obesity or is it a result of obesity? And so that's a very difficult question to address because it's not just a single microorganism like an infectious agent that you could infect another mouse with and see if it causes disease. In this case, we're curious about an entire ecosystem and what it can do. And so they performed a very clever experiment of a microbiota transplant where they took the microbiota from either lean mice or from obese mice, transferred those microbiotas into two different groups of recipient mice, and then just fed those two groups of mice the same thing and watched what happened. And the remarkable thing was the mice receiving the obese microbiota gained weight more rapidly. And so that was really the first evidence that the microbiota could be causal for something like obesity, this big host phenotype that was quite different than just gut health. And since that time, it's become apparent that our gut microbes are, sending chemicals into our bloodstream that circulate through our bodies and can influence, all aspects of our biology because our circulatory system goes everywhere in our body and carries these chemicals. And so now we're beginning to think about how can we use the microbiota, because it's malleable and we can change it, as a lever to tune different aspects of our biology, either treat disease or prevent disease.
That sounds fascinating. And how far along is that research? Or is that just in the very beginning phases, or are there some situations in which we know what to do to get a result?
There's one really great example of how microbiome reprogramming has been very successful in treating a disease, and that disease is C. difficile-associated colitis. So these are people that have an overgrowth of C. difficile in their gut, and it can cause terrible diarrhea. People die from this. It's really a difficult problem. And usually it was treated just through heavy doses of antibiotics in the hopes that you could bring down the C. difficile organism. And that was, somewhat successful, but not a great strategy. And then they found that if they just gave these people a microbiota transplant similar to what had been done in that mouse experiment, so just take a healthy donor stool and, provide that to the C. difficile infected person, that it could reboot their microbiota, and the cure rate for that was amazing, like basically, almost 100% cure rate. So that was one specific example of just rebooting somebody's microbiota and disease going away. Since then, there are a number of microbiota reprogramming or fecal microbiota transplant, FMT is how they call it, studies looking at if that strategy could be effective in dealing with a number of diseases, diabetes, metabolic syndrome, obesity, different autoimmune diseases. And so those studies are just starting. There's been some results that are promising, others not so much. And so there's a lot of work left to be done to figure out strategies to use microbiota reprogramming to cure disease, but it definitely is something that's very promising.
There's a condition that's really hard to treat. It's called inflammatory bowel disease. Things like ulcerative colitis, the drugs that we use are expensive these days, and sometimes they have side effects that are serious. What about this concept of reprogramming for bowel disorders like inflammatory bowel or even irritable bowel?
Yeah, I think that there are a lot of people that are excited about that. And there are, in the literature, there's definitely several trials underway and some indication that there is success with specific treatment regimens where fecal transplant is applied repeatedly. It's very different than the C. difficile situation where there's actually an infectious agent, a bacteria that's responsible for causing the inflammation, a single bacteria that can be eradicated through this fecal transplant. In the case with something like inflammatory bowel disease, where the host immune system and the barrier of the gut have kind of been thrown into a state of inflammation and being compromised, that it appears that it's more difficult to reprogram the gut into a healthy microbiota that can restore health. And so I think that what the field is learning is that there's going to need to be a variety of tools, perhaps a dietary reinforcement, perhaps defined communities of microbes rather than a crude fecal transplant, perhaps in combination with some of the conventional therapies, things like immunosuppressants that could stop the immune system from being inflammatory for a certain period of time while the system rebuilds and restores homeostasis. So I think that there's incredible promise and a lot of people looking into that, but the answers are definitely more difficult than C. difficile colitis.
One good analogy could be, if you think of like starting a garden and you throw a bunch of seeds on there, but then you don't water it, you don't fertilize it, like those seeds are not going to grow and form a lush garden. And so in many ways, this fecal transplant might be like that, in that you introduce these organisms, but you also have to introduce the foods that they like or create an environment that is helpful for them to then regrow. And that's something that I think the field is trying to figure out, all these factors to get a good community to really entrench in a person's gut and do all the benefit that they're capable of doing.
There's been a lot of talk about the donors, the healthy donors that are, if you like, submitting samples to repopulate the digestive tract. And we've heard recently about super poopers, that is to say, people who are super healthy and have a great diversity and really great bacteria in their gut. Are we starting to figure out, what kind of bacteria might be more productive than others?
Yeah, again, it's a great question, and I think as the field marches forward, we are getting a sense for what are kind of general principles of a healthy microbiota in terms of the membership, in terms of certain functional features, production of certain molecules that may restore the barrier. So I think we are starting to get a general idea of that, and also a general idea of some red flags in a microbiota, things that may be an indicator that there's inflammation occurring or other bad things happening. But the really difficult part in this field is, we mentioned the fecal transplant experiments that were done to cause mice to become obese. Those same experiments have been done to modify mouse behavior, to change the rate of Alzheimer's occurring. There's all of these phenotypic effects of changing the microbiota, but in almost all of these cases, it's very difficult to trace it back to a single molecule, a single molecular pathway, or a single species, or even a group of species. Instead, it's, a collection of hundreds of species that appear to do this, and it's hard to know, because of the complexity, what the important features are that underlie that. And it may not be reducible. It may be something that's an emergent property of this complex community. And so, I think the idea of screening somebody based on their health and based on, the certain features that appear to partition with a healthy population is kind of the best that we can go on now, but it's still not very specific and certainly not a fail-safe approach.
If behavior and cognitive function change with changing microbiota, how are the gut bacteria communicating with the brain?
Yeah, so we have some, plausible routes of communication, but again, the exact pathways and which ones are the major pathways are still widely debated in the field. So there's, our gut possesses a nervous system, the enteric nervous system, and that has a connection to our central nervous system. And so there have been experiments to show that neural connection is essential in communicating certain aspects of the physiology that's occurring in the gut to the central nervous system. So in some cases, it's routed through our nervous system. In other cases, there are molecules, little chemicals that the microbiota produces that get absorbed through the gut into the bloodstream, and then those circulate through the body, and some of those can cross the blood-brain barrier and enter the central nervous system. So that's yet another route. And then, a third route is that our gut is the most immune-rich tissue in our body. That is, the vast majority of immune cells in our body reside in our gut. And that makes a lot of sense because there's so much microbial life there. We need to be monitoring that and be educated about, what microbes are living in our gut from an immunological perspective. That immune system is not stationary. So the cells in the immune system, of course, circulate through the body. They work their way into tissues like the gut, interact with microbes or microbial products, and then they pick up and they go into the bloodstream and move through the body and then rehome to another tissue. And that's another way that you could imagine the gut microbiota interacting with the nervous system. And so there are all of these different routes that are in play and probably are, again, it's a really complex system, probably are working in concert in different ways to influence things like moods, behavior, neurodegeneration.
I'm fascinated that the gut has so much immune activity. I mean, I think back to the old hygiene hypothesis that is still evolving, the idea that if you grow up on a farm and you're exposed to lots of bacteria, you may be less sensitive to allergies and perhaps asthma. Can you go into a little more depth on this immune system that is basically dependent to a large extent on the health of our microbiota?
Yeah, so the immune system, I think, again, classically has been thought of as a way to fight germs. To, if a virus or a bacteria invades, it's the part of our biology that's there to kind of create war and to defend our human tissue. But I think the more nuanced and evolving view on this is that the immune system is actually just a microbial interaction interface. It's a way of perceiving the microbes, whether they're beneficial or disease-causing, and then kind of getting our body ready for, potential attacks, but also to deal with beneficial microbes as well and respond appropriately. And so there's no doubt that the immune system all over our body is tuned by the microbes that we have in our gut. And there have been, many experiments in animal models to show this, and now experiments in humans that are supporting it, that if you change the gut microbiota, you can fundamentally change how the immune system operates. So you can change how you fight off a viral infection in your lung by changing the gut microbiota. You can change the rate at which autoimmune disease progresses. For instance, if you have autoimmune disease in your central nervous system, you can actually influence the rate of that by changing the gut microbiota. And now there's many studies out showing that cancer patients that are enrolling in immunotherapy trials, these are trials where the drug is actually activating the immune system to fight the cancer, that the efficacy of those drugs can actually be influenced by gut microbiota composition. And so there's really this tremendous realm of new directions to go with tuning the immune system. Whether you're getting vaccinated or whether you're fighting an autoimmune disease, you may want to tune your immune system to a different set point, and the route to doing that may be through changing your gut microbiota.
I've been wondering where all the germs and fungi and viruses that end up in our gut come from. I mean, you've already described the fact that there are, hundreds of millions, maybe billions of bacteria in our digestive tracts? Well, how did we acquire them?
So many of our microbes early in life come from our mother, family members, and caregivers. Certainly when we're infants, if you're born through the birth canal, vaginal microbes are the first microbes you encounter. And then you gradually acquire other microbes from your mother and from the people around you. Because microbes are everywhere and the world is covered in a patina of microbes, that means that at a very low level, we're continually introducing microbes from somebody else's gut into our digestive tract. You can think about it as a low-grade fecal transplant that you're continually performing on yourself. And this isn't necessarily a bad thing to get exposed to more microbes that can increase your gut diversity. Whether those microbes stay in your gut will depend upon what you're eating, what the resident community already looks like. So, there's a complex array of factors, but gradually over the course of our life, we are being exposed to a wide array of gut microbes from the people around us.
You're listening to Dr. Justin Sonnenberg and Dr. Erica Sonnenberg from the Department of Microbiology and Immunology at the Stanford University School of Medicine. Their book is The Good Gut.
After the break, we'll learn how we can take care of our microbiota so it can take care of us.
We'll find out about the care and feeding of our microbiota. What does it like to eat?
You'll get some recommendations for breakfast, lunch, and dinner.
Find out how fermented foods can nourish our microbiome.
Are there strategies that can help if we need to take antibiotics?
You're listening to The People's Pharmacy with Joe and Terry Graydon.
Welcome back to The People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. If you'd like to purchase a CD of today's show or any other People's Pharmacy episode, you can call 800-732-2334. Today's show is number 1156. The number again, 800-732-2334, or you can find it online at peoplespharmacy.com. You can also download the free podcast from iTunes or from our web store. We invite you to consider writing a review.
The People's Pharmacy is brought to you in part by Verizana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome. Online at Verizana.com. That's Verisana.com.
Today we're discussing how our microbiota influences our health. In addition to digestive function, our resident collection of bacteria, viruses, and eukaryotes has an impact on our immune system and even our behavior and cognitive capacity. How should we take care of our microbiota so that it can take good care of us?
We're talking with Dr. Erica Sonnenberg and Dr. Justin Sonnenberg. She's a senior research scientist, and he's an associate professor in the Department of Microbiology and Immunology at the Stanford University School of Medicine. They're co-authors of The Good Gut, Taking Control of Your Weight, Your Mood, and Your Long-Term Health.
Erica, you've suggested that we need to take good care of our microbiota if we want them to take care of us. How do we do that?
Well, what the field has learned over the past decade of studying the microbiota is that the biggest lever that we have to control this community in our gut is through diet. So what we eat majorly influences not only the composition of the microbiota, but also what those bacteria are doing. And in a way, it makes sense, right? We're providing food in the habitat in which they live, our gut. And this is what we're eating is what, in turn, the microbes have access to as their food. And so what we choose to eat will influence what their options are for eating and their metabolism.
What do they like?
So one thing that people need to understand is that the majority of these microbes live in the most distal parts of our gut, so in the large intestine or colon. That's where the vast majority of these microbes are. And so what they like to eat is the food that actually reaches that part of our digestive system, which is specifically dietary fiber. So the human genome is actually terrible at its ability to digest dietary fiber. So when you eat food, things like the sugar or fat in that food gets absorbed relatively easily in our stomach and small intestine. But because our human genome doesn't encode much capacity for degrading dietary fiber, it largely remains untouched. And so it continues through our digestive system and reaches our large intestine where the majority of our microbes live. And this is the food that our microbes really love to eat, is dietary fiber, these complex carbohydrates that are found in plant material, fruits and vegetables, grains, nuts. All these things contain these complex carbohydrates that really fuel the microbes that live in our gut.
So if we were to join you for breakfast, lunch, and dinner, what would we be eating to encourage all of those microbiome bacteria to just dance for joy, thrive, the good ones, and maybe discourage the bad ones?
You know, I think part of that question is thinking about what we're confined to eating in the food system that we all live in, and whatever industrialized country. In the United States, we don't have access to a lot of the really fiber-rich foods that we ate long ago as ancient humans. And so if you visit, for instance, a hunter-gatherer society like the Hadza in Tanzania that are modern people, but they're kind of a continuation of humanity's ancient past, they eat on the order of 100 to 150 grams of dietary fiber per day. We as Americans on average eat about 15 to 20, so 7 to 10-fold less dietary fiber than we probably ate when we were evolving before industrialization. And so if we really could focus on what we should eat, it should be these fiber-rich tubers, not the potato French fries that we eat now from McDonald's. I think eating incredible berries that are not overly sweet but really high in dietary fiber, have thick skins. These are the types of foods that really humans should be eating, but of course that's not practical. These aren't foods that we can cultivate and distribute widely. And so given our situation and where we live, we really need to make deliberate, conscious decisions to find plants as the basic, soul of our diet that are high in dietary fiber. And so that's avoiding things like white rice or potatoes without the skins. These starchy vegetables do not have high dietary fiber, but instead whole grains, legumes, vegetables, nuts, and then some fruits, although a lot of fruits are very high in sugar. So I think, focusing on those plants that are known to have high dietary fiber is the only way to kind of come close to the amount of dietary fiber we probably should be eating.
Well, I'm wondering about breakfast because, a lot of people have eggs and maybe sausage for breakfast, or they're having, Wheaties or Cocoa Puffs or some kind of cereal. There are a lot of our listeners who are now gravitating towards smoothies that might have berries in them or even kale. What about a good breakfast? And then are we confined to salad for lunch, or are there some other things we might want to have?
Yeah, I think, breakfast is really important. And traditionally, American breakfasts seem to fall into like two categories of, like you said, the eggs and bacon, or the sugary cereal or pancakes or something is the other category. And if you look at the dietary fiber content of both of those types of breakfasts, they're really very bad. I mean, essentially nothing for your microbes. And at that point, your microbes, have gone the whole night with no food. Then you wake up for breakfast, you eat sugary or fatty food that doesn't have any dietary fiber. So that's, many more hours until lunch, until maybe lunch is the first time these microbes get any access to dietary fiber. And so I think breakfast is really important to really focus on getting a high fiber start to your day. And so smoothies are a great way, like you said, adding kale or berries. Other things that, we do at our house is put things like flaxseed meal or wheat germ, different things like that to try to boost fiber content. The other cool thing about smoothies is that it's a good way to start introducing fermented foods into your diet. So adding things like yogurt or kefir that have live microbes in there, you get sort of a double whammy of dietary fiber and beneficial microbes in the fermented food. Things like steel-cut oats, that has a really high fiber content. These whole grain cereals are a way of moving away from highly processed cereals into things that are more or less processed and more fiber content.
Now, I think most of our listeners know what yogurt is, but maybe not everybody knows what kefir is. Can you tell us, please? And do you put kefir on your steel-cut oats?
Yeah, absolutely. I'm a kefir junkie. I think it's, a wonderful way to start the day, whether you put it in your smoothie or, like you said, dump it onto oats or another, whole grain kind of muesli cereal or granola. So kefir essentially is a fermented milk product similar to yogurt, but most yogurts are made with a defined number of bacterial strains, kind of on the order of 4 to 6 different species of bacteria. And kefir instead is just this wild community, complex community of dozens of microbes that are working together to ferment the milk. And it results in typically something that is thick and drinkable as opposed to yogurt, which is, something that you would scoop with a spoon. I culture my own kefir at home. I actually culture 2 different types. You can order kefir grains on the internet and reconstitute them. They come dried. And it's a wonderfully easy way to have fermented foods as part of your daily diet because you don't have to heat the milk. It'll ferment at room temperature. So every morning I just strain my kefir grains. I drink the kefir that comes through and I put the grains that I've strained out into a new batch of milk in a jar, screw the lid on kind of loosely and let it sit overnight. And then I repeat the next day. So kefir, I think, is really a great thing that is, easy to do on your own at home.
What are some other fermented foods that people can eat that would nourish our microbiome?
One fermented food that is becoming very popular is kombucha. So that is similar to kefir in that it's an ill-defined community of microbes. So this SCOBY is a complex ecosystem of bacteria and yeast that ferment something like a tea or juice and create this drink that not only has the sugar reduced because these microbes have used those sugars for their fermentation, but also contains the living organisms from the culture. And so since it's a drink, it's pretty easy to, just get a couple servings of fermented foods just through drinking kombucha instead of, like juice or soda that are very high in sugar and don't offer any living organisms. The other popular ones are fermented vegetables like sauerkraut or kimchi if you like spicy food. Sometimes if we, make eggs, we'll sprinkle kimchi on top, and it's a way of adding fermented food but also adding some really interesting flavors and spice.
And, one other point there, just to emphasize what Erica said, really with fermented foods, there are at least 3 great benefits. One is the microbial life that's been growing and fermenting the food that you add to your digestive tract and increase diversity. The second is you're decreasing the glycemic load of that food because the microbes, as they ferment, are eating all of the simple sugars. And so they're really depleting sugar, which is why most of these foods don't taste so sweet. And therefore, when you eat that food, you're less likely to have something like a blood glucose spike because the glycemic index has been lowered. And then the third great thing is all of the microbial fermentation end products, which gives these foods their sour taste. Those are known to be also very beneficial for health. And so those 3 things together really make fermented foods something that we probably should be eating a lot more of. The thing that you have to be cautious of when you choose fermented foods, if you're not making it yourself, is that a lot of companies out there will try to mask that sour taste by adding back sugar at the end before they package it. And then in some cases, they even pasteurize it and can it, and then they're killing all the microbes. So if you want good fermented food, you really have to get it from a refrigerated section that shows that the microbes are alive and it has an expiration date on it. And then the other part is you really have to monitor and make sure that there's not a lot of added sugar that they've put back into either the yogurt or the kombucha to try to make it less sour and probably much less healthy.
There are times when we have to take antibiotics. Sometimes children have to take them for long periods of time. And we've also heard recently that colonoscopies, because you sort of quote-unquote clean out the lower digestive tract, may also impact our microbiota. How do we get back the good bacteria that we need besides nourishing them? What about probiotics? How would we know what to take? Which ones are going to be most beneficial?
Yeah, that's a very difficult question because of the marketplace is just flooded with different probiotics out there. And so the question is, how do you choose something and how do you choose something that's appropriate for a specific indication, whether it's trying to rebuild your community after antibiotic treatment or if you're dealing with something like constipation or for other reasons. And so we really recommend that people talk to their doctors about this, because if they have a specific indication for why they want to, take probiotics, there may be a study out there that shows a specific product is effective in dealing with that. But one of the big problems with the probiotic market is that it's not carefully regulated. And so there are a lot of products out there that are either mislabeled or contain only dead bacteria. And so it really comes to the buyer doing their diligence and there are specific seals that you can find on probiotics that shows that a third party has verified what is in the product, that there are live bacteria and that it agrees with what's listed on the label. But it's quite often a trial and error process for consumers where they basically, have to try different probiotics to see what agrees with them, what can ameliorate symptoms. Now, when it comes to building a community after you've taken antibiotics or had a bowel prep for a colonoscopy, there's just not a lot of great data out there to suggest that probiotics are having a huge effect or that there are some products that are superior to others. And so our general rule of thumb when somebody we know has to take antibiotics is to try to introduce dietary fiber back as you are able to into your diet to help the resident microbes regrow. And then fermented foods or, a yogurt, something like that there's quite a bit of data to show that those are beneficial in reducing things like diarrhea after antibiotics. So there's not a great specific answer, but just kind of these general rules of thumb that we know right now.
We are nearly out of time, and we're going to encourage everyone to read The Good Gut. It's a great book. But what do you want our listeners to take forward from this conversation?
I think the most important thing that's coming out of microbiome research right now is that we have this microbial component of our biology that we have to be aware of, that we have to care for, that can deteriorate over time. I mean, there's a lot of evidence right now that the industrialized lifestyle is not a friendly lifestyle for our gut microbial community. And so that means we can be more aware and do things in our life to protect and nurture and take care of this community, and that will come back to reward us in better health.
Dr. Justin Sonnenberg, Dr. Erica Sonnenberg, thank you so much for talking with us on the People's Pharmacy today.
It's been a pleasure. Thanks.
Yeah, thanks so much.
You've been listening to Dr. Justin Sonnenberg and Dr. Erica Sonnenberg. Dr. Justin Sonnenberg is an associate professor in the Department of Microbiology and Immunology at the Stanford School of Medicine. Dr. Erica Sonnenberg is a senior research scientist in the Department of Microbiology and Immunology at the Stanford School of Medicine. She studies the role of diet on the human intestinal microbiota. They're co-authors of The Good Gut, Taking Care of Your Weight, Your Mood, and Your Long-Term Health.
Lynn Siegel produced today's show. Al Wodarski engineered. Dave Graydon edits our interviews.
The People's Pharmacy is produced at the studios of North Carolina Public Radio, WUNC.
The People's Pharmacy theme music is by B.J. Liederman.
The People's Pharmacy is brought to you in part by Verazana, an analytical laboratory providing home health tests for hormones, gut health, and the microbiome. Online at verisana.com.
If you'd like to buy a CD of today's show or any other People's Pharmacy episode, you can call 800-732-2334. Today's show is number 1156. That number again, 800-732-2334, or find it online at peoplespharmacy.com.
When you visit our site, you can share your thoughts about today's show. Have you given any thought previously to the care and feeding of your microbiota? The Sonnenbergs have a number of suggestions. We have a link to their lab website from our site.
At peoplespharmacy.com, you can sign up for our free online newsletter or subscribe to the free podcast of the show. You'll never miss another episode, and you can share it with a friend.
And when you sign up for the newsletter, you'll get our free e-guide to favored home remedies. In Durham, North Carolina, I'm Joe Graydon.
And I'm Terry Graydon. Thank you for listening. Please join us again next week. Thank you for listening to the People's Pharmacy Podcast. It's an honor and a pleasure to bring you our award-winning program week in and week out. But producing and distributing this show as a free podcast takes time and costs money.
If you like what we do and you'd like to help us continue to produce high-quality, independent healthcare journalism, please consider chipping in.
All you have to do is go to peoplespharmacy.com/donate.
Whether it's just one time or a monthly donation, you can be part of the team that makes this show possible. Thank you for your continued loyalty and support. We couldn't make our show without you.